overview of an example of genetic engineering process from the first step until you get your product, for example, insulin. So you'll get an overview. So step 1 is you want to obtain the wanted gene, right? So you already know there are 3 ways to obtain the wanted gene. From DNA to DNA, from mRNA to DNA, from protein to DNA, 3 ways. And the best way is to obtain the mRNA convert to DNA. So in this case here, beta cell in the pancreas will actually secrete insulin. So when it expresses the insulin gene, a lot of mRNA will be produced. So we will isolate the mRNA. From mRNA, we will convert it into double-stranded cDNA. Okay, so this obtain the wanted gene. Then from the bacterium, we must obtain the plasmid because we're going to use it as a vector. So again, you isolate it. So now we've got 2 different types of DNA. So we use the same restriction enzyme to cut both the DNA. And then you put both the DNA together in a test tube and allow them to fuse. And then they will fuse and form recombinant DNA. So we need to use enzymes to do that. Okay. So after that, don't forget in a test tube, not only you got recombinant DNA, you also got plasmid and insulin gene that did not bind together. So you got 3 products. Now, this test tube got 3 products, you add it into the bacteria. So among all the bacteria, only 1%, only 1% of your total bacteria will take up this recombinant, will take up the plasmid. Can you see? I draw 4 bacteria, only 1. So actually it's 1% only will take up. So after 1% take up, of course, you have to, the next step, select, right? Which bacteria has been transformed. Transform means take up the foreign DNA. So this process of selection, that's where we use the marker. Marker means, remember, you mark the one that has taken up the gene. Marker, right? So you have to use the marker gene to do that. You know how to do it later on. So after you select the transformed bacteria, then we can clone the bacteria. Clone means what? Make identical copy, isn't it? Right? Clone means make identical copies. So in this case here, when you want to clone copies of what? Copies of the gene, actually. We want to make multiple copies of the gene. In order to do that, we must make the bacteria to divide to replicate the plasmid. Because when they replicate the plasmid, they replicate the gene. So that means to say that we must allow the bacteria to divide, right? In order to clone the gene, the plasmid must replicate. In order for plasmid to replicate, that means the bacteria must divide. Right? From 1 cell it becomes 2 cells, 2 cells becomes 4, 4 becomes 8, and so on. So it will double every time. Okay, that means to say that in order for bacteria to divide, you must actually culture them. Culture means to grow them. Grow them in a suitable nutrient medium. Make sure you write all this down in your notes. Okay, grow them in nutrient medium. Like you want to grow the baby, you feed them with milk, right? Because milk contains what? Protein, carbohydrate, lipids. You want to grow the bacteria, you also must feed them with carbohydrate, lipids, proteins, and some mineral ions. So these are called the nutrient medium. So therefore, bacteria in a suitable nutrient medium, suitable temperature, optimum temperature, it will divide. So when divide, it will replicate the plasmid. When you replicate the plasmid, you clone the insulin gene. Then after that, when the bacteria express the gene, that means to say it will then synthesize the insulin. Then you get your end product of this genetic engineering. Right? Interesting, isn't it? Right? From the most basic knowledge that you learn about DNA and gene expression, you can design this process so that nowadays, right? You know, in the past, insulin is very difficult to get. It's very expensive because they have to get it from the pancreas of slaughtered animals. You know, you imagine for every animal we slaughter, how much we only get 1 pancreas, and from 1 pancreas, how much insulin we can get? It's very little, isn't it? And on top of that, there could be infection because there could be bacteria there. And then animal insulin is not similar to human insulin. So therefore, its effectiveness in the homeostasis is not as good as our own human insulin. So all this got problems. So now this problem is resolved because we have used bacteria to synthesize human insulin in mass production. Because of mass production is easily available and it's cheaper. So therefore, then this insulin can be supplied to those people with diabetes. and they can have continuous supply all the time. So that is how powerful it is when you apply the basic knowledge that you know, design something so that you produce products and processes that are very useful for the mankind. And not only for the mankind, probably for the plants and animals and the planet. Okay, so small little things, when you do it well, will actually bring benefit to many people. Same thing, small little things, when you don't do it well, it's disaster for many people. It's the same, right? It's opposite. Okay, so we just take care of our action, thought, action, speech, and action. Then you make a big difference for ourselves and also for people around us lah. That is always applicable throughout the entire life. Okay. Okay. So I'm not going to go through the step because we already this is just a general overview for you to know, and I've actually touched on some of the steps here, but because in the PowerPoint slide I'm trying to compile everything together, so I put all the steps again, but actually you've learned it already. Okay, so therefore we'll continue to learn the steps now. Okay, let's look at the words before we continue. So let's put the diagram into words. So the 5 steps. The 5 steps in genetic engineering to synthesize human insulin. Now this is an example of GE, specific for producing human insulin only. Okay, because different product we use different method. This only for insulin that has been used, this process. So first we obtain the wanted gene. Now the wanted gene is obtained by this process from mRNA to cDNA. Now after we obtain the wanted gene, right, the quantity of this DNA is very little only. So if you want to insert this DNA to many bacteria, you have to first multiply this DNA, right? So there's another step that we need to do to multiply this DNA. So this step is called amplification. Amplify means to make it more. So what do? We clone the DNA. So later on, you see how to clone the DNA using this technique called polymerase chain reaction, in short PCR. Okay, then after that, you have to process the cDNA by adding a leg operon to it. So you need to add a leg operon in front or upstream of the insulin gene. Later on, you know why. Actually, now you also know why already, right? So after that, your DNA is ready already. Then you insert the DNA into the plasmid vector, right, to form recombinant DNA molecule. Then transformation means you insert the recombinant DNA to the bacteria. Then you select the bacteria that has been transformed using a marker gene. Antibiotic resistant gene has been used in the past, but now we do not use antibiotic resistant gene already. Then after that, after you select transformed bacteria, you clone and express the insulin gene in this bacteria, and then you go and harvest the human insulin. Okay, so what you're going to continue to touch on is actually amplification, adding of leg operon, as well as the marker. Where's my marker here? Okay. Right, so let's look at amplification first. So we learn by following this sequence. Right?